Hi everyone, welcome to our second video on the plasma membrane. And in this video we're going to look at the processes of membrane transport. Now, before we go into the details of how molecules can be moved across the plasma membrane, I'd like you to take a look at this list of substances and decide which you think will be able to move through the plasma membrane unaided. Feel free to pause the video for a moment if you need to. Okay, let's have a look. Did your list match what just happened? Why do you think some of them were able to cross while others weren't? The answer comes down to the nature of the substances. Those that are non-polar will be able to cross the membrane, while polar or charged substances will not. This is due to the hydrophobic fatty acids of the phospholipids. If a substance is polar or charged, it won't mix with the fatty acids, and so won't be able to get through the core of the membrane without assistance. We're going to look at a few different mechanisms of transport across the plasma membrane, and we're going to start with the simplest, diffusion. This is a type of passive transport in that the cell does not need to expend any energy to make this happen. The energy is provided by a concentration gradient. The molecules are all grouped together in one place, making the concentration high, and they'll tend to move towards where the concentration is low. It's important to remember that the movement of each molecule is random, but the net movement will tend towards the molecules spreading out over time. If we consider our green spheres here, this means that over time they will tend to move towards the other side of the membrane until the concentration is evened out on both sides. So, what happens if we bring in a second type of molecule? How do you think our blue triangles will affect things? Well, the simple answer is that they won't. Each type of substance will move down its own gradient, regardless the direction of movement of the other. Now, as we saw at the beginning, substances cannot diffuse directly through the plasma membrane due to being polar or charged, and as such will require the help of a transport protein to get them across the hydrophobic core. This could take the form of a channel, which can open to provide a tunnel or corridor for the substance. This is pretty common with substances like ions, where each type of ion will have its own specific channel, which can be opened or closed in response to environmental conditions. Other substances, like glucose for example, will move across the membrane with the help of a carrier protein. These will undergo a shape change after binding the molecule, and will then shuttle it from one side of the membrane to the other. Now, even though there are proteins involved in these processes, this is still diffusion, as the transported substances are still moving down their concentration gradients, and so the cell still doesn't need to use any energy to make this happen. As such, this is referred to as facilitated diffusion, literally diffusion with help. The third type of passive transport that we're going to look at is osmosis, which you can think of as diffusion of water. Since water is polar, it moves very slowly through the plasma membrane unaided, but moves very quickly through water channels called aquaporins. Water will move in the opposite direction of solutes. If we consider our green spheres as solutes, you can see there are more on one side of the membrane than the other. And where the solute concentration is high, the water concentration is low. The water molecules will move through the aquaporin towards the higher solute concentration until the concentrations are equal on both sides. It's important to note the difference between concentration and volume here. Osmotic pressure can lead to the volume on one side of the membrane being higher than the other, as is required to make the concentrations of solute equal on both sides. Our final type of transport that we're going to look at is active transport. And this one, as the name suggests, does require energy expenditure from the cell, typically in the form of ATP. In this example, we're going to consider the sodium-potassium pump, which is one of the most important active transport systems in animal cells. In this, the sodium, as demonstrated by the green diamonds, is being pumped out of the cell, moving it from where its concentration is low and adding it to where its concentration is already high. At the same time, we're going to be moving potassium ions, the purple circles, into the cell, again moving them against their own concentration gradient. In this process, the pump will first of all pick up 3 sodium ions, and will use energy from ATP hydrolysis to change shape and push the sodium out of the cell. In its new conformation, the pump can pick up 2 potassium ions, causing it to revert back to its starting shape and release the potassium into the cell. As we mentioned, this pump is really important in animal cells, such as for maintaining the function of the nervous system. Other forms of active transport are also important in functions like absorption of nutrients in the small intestine and maintaining water balance between cells and extracellular fluid, which we'll look at more in another video. Thanks for watching, and please make sure to check out the other videos in this section.